Okay, so I'm from the Technion in Israel, that's in Haifa. We do both computational and experimental work. And it's a shame we don't have a whiteboard here, because for me the whiteboard is the extension of my mind. So I have a big whiteboard in my office, and I like to take a photo sometimes and see how, all the different kind of things mix up together. So this is one example. So you can see essentially everything we do here. I don't know how well you can see, but at the top you see that's DNA. So we deal with DNA. So we try to think about molecular mechanisms and figure them out. We do also mathematical models. We do method development, experimental method development, and computational method development. So everything here mixed together, and you can summarize it with these two icons, a computer and a Eppendorf. So that's kind of, we do lots of very different things. Of course, I won't be able to talk about everything today. Now, since I don't know how much time it's going to take, I'm going to start with the acknowledgements. So I'm just thanking current and past lab members. This is the Faculty of Medicine, actually, this ivory tower. And collaborators on stuff I will show. Okay, hopefully we won't have a very bad connection. Okay, so before this, we start, one last thing. Yesterday I felt bad that everyone was presenting their hobbies, and I have a twin brother and all this stuff. So briefly, I also have a twin brother, actually. And that's me on the left. He's on the right. I suspect he's maybe Indiana Jones. This is when we were trekking in the jungle in Guatemala to El Mirador. Recommended. Anyway, so now we can start. Okay, so we're going to go a long way back in time to Fleming, who coined the term chromatin. And back then, biologists need to know how to draw. So they have these beautiful drawings. And what he saw was all these interesting structures going on right inside the nucleus. And we know, of course, that's cell cycle, right? As I said, he came up with the term chromatin. Most of the times, it looked like there's nothing interesting going on, right? This is an interface. And this was often likened to spaghetti. Actually, it's not spaghetti. So this is the first thing I want you to note and to remember. So in textbook, you usually see these drawings of a folding, and you all know probably the basic layer of folding around histones to form nucleosomes. That's very, very small scale, right? About 150 base pairs, while the genome is very large, right? Human genome, how many base pairs?
3.2 billion.
Great. Perfect. It's nice that, more computational people or people who deal with data a lot know this, very easily. Sometimes when I ask biologists, graduate students, no one knows the size of the genome, which is sometimes embarrassing. Anyway, so beyond this level of packing, we have all kind of drawings of the solenoid and all kind of spaghetti and noodle stuff. It's actually probably wrong. So we're going to mainly deal with these kind of structures. And of course, you know that structure is also related to functions. So it's not surprising that it's not just a random mess. So I guess two classical examples. One is enhancer-promoter interaction that can occur at very large distances. How large?
Hundreds of thousands of nucleotides.
Right, even megabases, even a few megabases. Yeah, so it can work from really, really far. If you think, for example, a yeast chromosome is a few 100,000 or maybe a megabase. Okay, so this is quite long. And of course, there's also levels of regulation which are more smaller scale, like genome accessibility. Okay? Good. So the high-level questions that we ask, and many people in the field, very briefly, you can think of it as if you have the DNA sequence. This kind of dictates binding of different types of molecules. You get this molecular landscape of binding. And beyond that, you have maybe epigenetic information also added therein. This somehow is related to genome structure, and this is somehow related to function. So the first thing that people try to do is just describe the 3D organization of the genome. And we're mainly going to talk today about genomic methods because we're dealing with sequencing and describing how it changes along processes and so on. The other thing is kind of going upstream and asking what specifies this. Okay, so there's some information flowing in and specifying it. And the other is kind of downstream, what does it do? Okay, so we're going to talk a little bit about these things. So the roadmap for today is as follows. So we're going to talk, first of all, a little bit about the technology of measuring the 3D genome. Since you know it, I guess it's going to be fast. And then most of this part of the talk is going to be talking about the different things that we see in interaction maps, from very large scale to very small scale. Hopefully you will learn some new things. Then we'll talk briefly about how we can use this type of data in the context of a genome assembly and identifying structural variation. And then we're going to have this hands-on part, which is going to be pretty simple. It's basically going to be how you look at the data. I'm trying to give you a feel, kind of the basic processing, and most of the time we'll be just working with a notebook and using tools to look at these different types of patterns. Okay, so pretty basic stuff. And if we have time, hopefully we will, I want to talk a little bit about how we try to use mathematical models to understand the 3D genome. Okay, so I'm not sure yet actually what we will do first, but let's start and see where we get. Something important. So since I wasn't sure what the backgrounds here are, this is different focuses and so on, please feel free to ask questions, stop. There's no such thing as a bad question. Okay, so let's make it lively and questions and discussion. Okay. And I'll try to sprinkle in some research in the middle. Okay? Okay. Okay, so as you know, this was initially looked at with microscopy. Microscopy is still a very big thing. Unfortunately, we won't deal that much with microscopy here because, again, the focus is next-generation sequencing. But with the introduction of next-generation sequencing, as you know, many fields benefited from it. One is chromatin organization. Okay, so the basic experiment which was used to interrogate chromatin, and this is even before sequencing came into play, is something called 3C, chromosome conformation capture. This was developed by Job Decker about 20 years ago. And the idea is as follows. So you take a population of cells and you want to learn about proximity of loci in space. So the idea is to translate this type of information of proximity in space into something that you can measure by sequencing. Although here it wasn't with sequencing, I'll explain it in a minute. Okay, but this is generally the idea, right? All these sequencing experiments, you're trying to translate something so the output would be sequencing information. Okay, so it works like this. So you take a population of cells, you cross-link it with formaldehyde, then you cut it with a restriction enzyme. Okay, in this case it's a 6-cutter. We've since improved this a little bit. And then you're left with these molecules, which are, where's the mouse? Which are attached together. Okay, and you ligate them together. and then you purify. And now you end up with DNA, so this is just DNA. And you end up with molecules that didn't appear originally in the genome. So it's a chimeric molecule. The blue came from one part, the red came from another part. If we map it back to the genome, now we know that in one of the cells, these were next to each other. Okay, so you have this library of many, many such molecules, and you want to interrogate it. Okay, so originally this was interrogated by PCR. So you could ask about couples or pairs of loci, see how frequently they interact, okay? So remember, we're looking here at a population of cells. So the question is not whether they interact, yes or no, it's how frequently they interact. In what fraction of cells do they interact? And since then, there have been many ways of kind of interrogating the output, okay? So originally, it was kind of 1 versus 1, but there were other, since developments, it moved on. So you may be familiar with some of these. So 4C is one locus versus the rest of the genome, and you can use many versus many. And today we're going to mainly focus on the all versus all, which is mainly high C. We'll talk a little bit also about micro C, which is essentially some version of high C. Okay? So what's the deal with high C? So I guess you said you are familiar with high C. So why isn't high C just taking the output of this and sequencing it? Why wouldn't that work? Right? So you could say high C, okay, let's just take this and sequence it. Right? We're done. That's actually not high C. Any idea?
Well, you need, I think, to have isolated rows, capture complex.
Okay. So the thing is, you're right that the experiment, this is just a cartoon. So in the experiment, actually what happens is that the ligation is not perfect. It's far from perfect. Okay? So most molecules don't ligate together. Okay? In addition, there's another step here. If you think about the sizes, so Hint3 cuts every 6 base pairs. Right? So think how frequently you would have these sites. Okay, so it's 4 to the power of 6, 2 to the power of 12, right? That's about 4,000. Okay, so you're not going to get pretty large fragments, right? So you need to break them. Okay, now if you break a fragment like this, only the center part is going to be informative, right? If you break a part from here or from here, it's not going to be informative. Okay, so what happens if you sequence everything, you're going to get a very, very large fraction of molecules, which doesn't tell you anything. So it's going to be a huge waste of money. Okay? So this was why people, this was also developed in Jörg Decker's lab in collaboration with Erez Lieberman. And this is the updated version. So what happens here is you start the same thing, you cross-link, you cut, and now you fill the end with nucleotides that have biotin on them. And then you ligate them together, and you continue, and then what you can do is you can pull them down with streptavidin beads and capture only the informative ones, or mainly the informative ones. Okay? So this is the additional trick you need to do high C. Okay, so originally this was done, as I said, with a 6 cutter, so a 6 base pair recognition site. This was the first version in 2009. The second versions had an improvement. So first of all, they moved to a 4 cutter. Okay, so these sites, so the enzyme is usually DPN2 or MBO1, doesn't really matter. But the sites are going to be more frequent, okay? So it allows you to get to a better resolution, okay? Because remember that in the end, you're saying this fragment was next to this fragment. If they're small, you can get a better resolution. So this was one thing. The other thing, which sounds kind of technical and uninteresting, but it made a huge difference, was just the fact that originally, in the original high C, you'd open the nuclei completely. And the idea was originally they hoped that only molecules that are held together by formaldehyde would ligate together. So you don't get background signal. Okay? But it turned out that it works better if you actually don't disrupt the nucleus, you keep it. And this is sometimes called the nucleus in situ, you will see also. And when you do this, the data is much higher quality. So actually, all these parts are done within the nucleus. You actually insert the enzymes into the nucleus. And this is how it's done. This is the second version. If you're actually interested in doing, by any chance, Hi-C by yourselves, I'll also just briefly mention a preprint that we put up. I don't know if you can see it. It's an improvement of the protocol. It's called Fly Hi-C. We shortened it from 5 to 2 days. We reduced the reagents about 5-fold. And you can do it, instead of doing it on millions of cells, you can do it on tens of thousands of cells. So it's quite easy. The protocol is there and so on. Okay. So this is how the experiment works. You get this library of interactions. Now I'll show you. This is, you all know it, so I'll show it kind of very fast. So you're building now a heat map, essentially, where on both axes you have the genomic coordinates. So here you have a genome of size 7, okay? And if you have interaction between this blue and green, which map to positions 1 and 3, you're going to write a 1 here. And because the matrix is going to be symmetric around the diagonal, you're going to write a 1 here.
Sorry, can I ask a slightly technical question?
Yeah.
How do you put it in the nucleus?
So the nucleus is going to be slightly perturbed, so it's not going to be intact, but but most of it is held together. It's not that it completely bursts. Yeah. Okay, so essentially what you're doing is you're actually filling this up with reads, right? So you're filling it up, and you're going to color it like a heat map, right? And then you can see how frequently this is going to be the color scheme that we're going to use. So dark is going to be a lot of interactions. Down here is going to be a few interactions. Now, though, I'm showing you kind of this very simple toy example. Just now, please don't be shy. If you don't understand this, you want me to explain it again or something like that, this is what we're going to be looking at for 3 hours. Okay? So please don't be shy. I know at first it can be a little intimidating what these colors mean, because we're going to see all kind of patterns. Okay? So just to orient you, for example, if we look close to the diagonal, okay? So the diagonal is going to be here, the interaction between 1 and 1, or 2 and 2, right? Here, close to the diagonal, the first diagonal is going to be the interaction between 1 and 2, so they're nearby on the genome, okay? If you're going back here, far away, this is the interaction between the beginning and the end, okay? So things near the diagonal are interactions between loci which are close to each other on the genome, okay? Good.
Sorry, and what's the scale? Like 1, 2, 3, 4, 5, 6, 7? It's like, it's not, it's like how large?
Yeah, so right now, this is, I didn't really say what the scale is, right? But you could think that if the smallest thing that you can measure is the restriction fragment, right? Then this could be there are 7 restriction fragments in the genome. The first is interacting with the second or something like that.
So that means it's not linear.
In this case, it's not linear, but we're going to get to this in a minute, okay? A little bit into the details. Okay. Let's continue. Good. Okay. So that was a toy. Now, as you know, before we look at real data, so you have this, right? You can look in Google Earth. You can zoom in. Here, you're going to zoom in on Haifa and find the building of the Faculty of Medicine where I work. Oh, this is annoying. Okay. Yeah, so this is the building. Different scales, you're going to see many different things, right? So the same is with Hi-C, okay? So again, you need to get a sense of the scale. Think about the size of a chromosome, okay? That's a few hundred million base pairs. Size of a nucleosome is, let's say, 150 base pairs, so that's more than 1 million, okay, fold. So you have lots of different scales, and you're going to see there are lots of different patterns, okay? So now we're looking at kind of the entire genome, but move in, and you see, squares and rectangles and stripes and dots and all kind of interesting things. You can even zoom in more and find interesting things. Okay? Good. So basically, what I want to do is kind of go through these different things and tell you a little bit about them. So now we're getting to this thing with the numbers which were mentioned. Okay, so this is kind of technical, but it's important. Okay, so typical sequencing experiments, you just sequence a few tens of millions of reads or something like that. You're fine. If you have better coverage, then fine. But here it's a little different. Okay, so what's different? So the human genome size, we said it's 3 billion base pairs. Now, we have a restriction enzyme that cuts 4 base pair sites, okay? So you can calculate the restriction fragment length, let's say, will be something like 400 base pairs, okay? So this is the highest resolution you can hope to measure, right? Now, let's see if you can actually get to this resolution and what the implications are, okay? So how many restriction fragments do you have? Okay, so you take the size of the genome, you divide it by the size of the fragments. How many fragments do you have? You have about 7.5 million. Okay? Now, what's going to be the size of the matrix? Okay? So it's 5.6 times 10 to the power of 13. Okay? So this is a very, very big matrix. Okay? So you have millions by millions. So the possible interaction space, are we just going to hope for the best? I hope so. Yes, okay. So this is a very, very big matrix, right? So this is all possible interactions. Now, what we're looking for is patterns. What is a pattern? A pattern is an interaction which repeats in several cells. Okay, so think about how many reads you would need just to fill this matrix, okay? How many reads do you have, for example?
Can you repeat again what were the interaction space?
Yeah, so the interaction space is like the size of the matrix, right? Because you have, essentially you have 7.5 million possible, it's like loci in the genome, it's the restriction fragments. And each one can meet with all the others. So all the possibilities is more than 10 to the power of 13. So it's huge, right? So let's say you sequence a lot, you have a ton of money, okay? So you sequence, let's say, 1 billion reads, okay? That's quite expensive. That's 10 to the power of a 9th, okay? So you get the context of that here we have a real issue with sequencing depth, okay? Good. Okay. I won't say anything about the cells per experiment, but let's jump ahead a little bit. So the point is that the interaction space is undersampled. Okay, so the point I was trying to make is that we have a problem with the sequencing depth, okay? So it seems like it's going to be, okay, we have no chance, we can't do anything, but actually we can. So what do we do? So first of all, the interaction map is not uniform, okay? So there are some places where there are a lot of reads, and that even if you sequence roughly shallow, so even if you sequence tens of millions, you can see some things, okay? But you have to realize that the stuff you see depends a lot on the amount that you sequence. Basically, the more the sequence, the more you will see, okay? Okay, so what are the solutions? So first of all, one is to reduce the interaction space. How do you reduce the interaction space? So you can try to measure just part of the genome, okay? Or go work on bacteria. It's also really good. I recommend it, and you don't have these problems, okay? You can sequence more, but there's a limit to that. And another thing that you do is you bin the data. Okay, and now this gets to what was asked here. So what you usually do is you take now, instead of looking at one fragment, you're taking a window, let's say 10 kb, and you're counting all the interactions within that window, okay? And the size of the bin will affect the resolution that you look at, okay? So this is something that we're going to see. This is a little bit challenging with Haifa. When you look at the data, you need to look at it in different bin sizes. Okay, and this affects what you can see. I'll show you this. Okay, so practically what people usually sequence is hundreds of millions, sometimes billions, depending on what you want to see. So it's important in advance to know what you're looking for. Okay, good. Okay, so before we actually get to the patterns, I see this goes a little slow, but that's okay. This is really important to remember. Two things. First is, this story about the blind man feeding the elephant, right? And everyone thinks it's something else, right? Someone feels the leg, they think it's a tree trunk, and so on. The trunk, they think it's a snake. So the problem is that we're a little like the blind man here, right? Because we can't actually see the structure. We're just measuring these proximities, and we're measuring it across the entire population. Okay? And I'm going to show you some examples. I'm going to show you at least one example where I'm going to later kind of give you a glimpse of what the elephant really looks like, and you're going to see that we weren't able to see this in terms of interaction. Okay, so this is one thing. Another complication is this story, the one about the statistician that, so he drowned in a lake that the average depth was 30 centimeters. Okay? Right? So this is also a problem, that we're looking at the average. Right? We're looking at an average, and what you will see, this can be very, very misleading. It was also misleading actually to many people in the field, because you see a pattern, you try to think what kind of structure that represents. The natural thing is to think, in all cells it looks something like this, but it could be that in none of the cells it actually looks like the pattern. Okay, so it's probabilistic. And if you think about it, DNA it's just like, essentially if you take just naked DNA, it's like a random polymer. Okay, it just can move, it can do whatever it wants. This is really different from protein structure, where the protein itself has, the backbone and dihedral angles and all these physical constraints. In DNA, what gives you the constraints is the molecules that bind the DNA. Okay, so you have a completely kind of flexible thing, and then you have constraints in some places. So what it means when you look in different cells in the population, there's going to be huge amount of stochasticity. They're going to be very, very different from each other, except for specific places and specific things. And these are the patterns that we're looking at. Okay, so now we can start. Okay, so the first thing that you see when you look at the Haifa map is something like this. So at the top and bottom now you see the numbers of the chromosomes, and it's really obvious that you see these strong squares here, and the rest looks kind of uninteresting, right? So what does that mean?
That the chromosomes that are close to each other have more interactions. Oh well, no, that the line is because you are comparing the chromosome with the same chromosome.
Right. Okay, good. So each chromosome, if you look at this, you're going to say, well, the chromosomes tend to interact mainly with themselves, not that much between chromosomes. Okay? So what I'll show you, it's kind of half true. Again, you have this thing of population averaging. Okay? The fact that we're looking at the population also plays a part of it. Okay? So if you look between the chromosomes, it really usually looks like this. You don't see anything interesting. So most of the people actually really focus on what's in here. I'll tell you something interesting about that in a minute. Okay, so when you actually look at chromosomes in interphase, so this is from a simple experiment that we do in the lab, that you can, it's a DNA fish, so you put fluorescent probes on the chromosome. This is in interphase, and you see that the chromosomes are actually not kind of spread completely in the nucleus, but you can see that they're kind of in their own volumes. They're not compact like in mitosis, but they're still in their own volumes. And people have already found this. And you can see that chromosomes, even in interphase, are kind of occupy their own volumes. Okay? And these are called chromosome territories, these things. So first of all, you see that they're not really completely mixed. Okay? And this makes sense in terms of what you see also in the map, right? However, now if you look at, before the map. So this is one thing. The other thing is this, that if you look at specific chromosomes, so you have these territories, but now you look at their position in the nucleus, okay? So let's say the green and the yellow. So it turns out that these positions are highly stochastic. So if you look in one nucleus, chromosome 1 can be next to 2. In the other nucleus cell, it's completely in the same state, everything the same, but now 1 is on one side of the nucleus, 2 is on the other side, okay? So they can be very, very stochastic, okay? What is conserved sometimes is something called the radial positioning. So that's, if you take a nucleus and just look at how far a chromosome is in terms of the radius, how far it is from the nuclear lamina, then you will see that this is sometimes representative of the chromosome, okay? And you find that short chromosomes, you can also see it a bit here. This is just interaction on the chromosome level. And actually you can see that short chromosomes tend to interact more, and they're actually more in the center of the nucleus. Large chromosomes tend to be more in the periphery. Okay, so it's kind of stochastic, but there is something that is still maintained. Okay, and actually this stochasticity, this is a major part of what you see in the map. Why? Because if 1 is sometimes next to 2, okay, but sometimes next to 3, sometimes next to 4, and so on, now when you average these together, it seems like it's not interacting with any of these. But if you go to a specific nucleus, you're actually going to see that 1 in this nucleus, 1 is next to 2. Okay? And this one, 1 is next to 5. Okay? So it's an averaging effect. And in fact, you can see this in single-cell Haifa, which I wanted to mention really briefly. So this is from over 10 years ago. Single-cell Haifa has developed since quite a lot, but still it's quite technically challenging to do. So don't expect to succeed in it. I'm not trying to depress you. We tried it for 2 years and it was hell, so don't waste your money on it. Anyway, so the idea how people usually do single-cell Haifa is because everything is in the nucleus. Essentially, this allows you to do the experiment on bulk and then separate the nuclei and then tag them separately. This is the idea. Okay, so if you look within now individual cells, so now each one here is a cell. Okay, and you're looking at the interactions of chromosome 2 in each of these cells. Okay, so first of all, you see chromosome 2 always tends to interact with itself. Oh, sorry, I'm not. Okay, so chromosome 2 first of all interacts with itself, but also sometimes you see, like in this cell, 2 is maybe next to this part of 7 and this part of 11 and so on, and this can change. Okay. So when you look at the population, it looks like there's nothing there. Okay, good. So now, okay, see we're a little short on time, so I'll tell you this pretty briefly. Just I wanted to sprinkle in some research. So usually, if you hear about Haifa, many people will tell you all the interesting stuff is within the chromosome. But we actually, I never like these assumptions, so we try to look for interactions between chromosomes which might be functional. Okay? So I'm just going to go ahead and not really tell you all the details. Essentially what we did is we looked at interactions between regions of different chromosomes, and we crossed this with data of the functional annotations of the genes within these two regions. So we looked for regions essentially that both have similar functionality in terms of the genes and also have strong interaction between them. And I'll show you one kind of very interesting example which we found and followed up since. I won't tell you the whole story, but actually the histone genes in mammals, they're encoded in 4 clusters. So this is in mouse, and you have several copies of these genes in 4 places on different chromosomes, okay? And these genes need to be highly coordinated, because when do you need to synthesize histones? When you replicate your DNA, right? You need more histones, right? You replicate your DNA, you generate a lot of histones. And it turns out that when you know where to look, you find very clear interactions, okay? So this is an interchromosomal interaction, very strong. Okay, this is in mouse ES cells between histone cluster 1 at the top and histone cluster 2. And just to be short, I'm going to skip this, but we also started to do microscopy to validate this. This is also a form of DNA fish, where we tag the loci fluorescently. You can see this is histone cluster 1 in pink, histone cluster 2 in green, and we also put a control locus, which is next to histone cluster 1. We wanted to see that these co-localize, but that this is specific. Not just that the chromosomes were next to each other, but it's specific to these regions. And you can see here these pairs. In this case, it's a triploid, so you see 3 pairs of each. So the orange, the control, is always next to histone cluster 1. And in this case, you have a co-localization of these. Okay? So the point is just, so we don't know yet what it's, you can ask me about it later. We have follow-up. We don't know the function yet of this thing. But be a skeptic. Okay? So if people tell you there's nothing there, ask, how do you know? Okay? And usually it's because people didn't look hard enough. Yes?
Just one question. Do you think, because usually I thought that interchromosomal, it's more like about fixation, different fixation between the artifacts and stuff. Do you see some differences if you try different fixations?
So we didn't play that much with the different fixations, but I did see that other, I know that other versions, like I think micro-C, so for example in micro-C I think you don't see these that well, these interaction. So the details are important. I think it could be that different cross-linkers make it easier or more difficult to see this. Definitely, yes.
I guess that the diameter is also important in the interactions, so it's a different method.
Right. So yes, and the question is how they actually get physically connected to one another through cross-linking. I didn't want to get into this, but actually cross-linking only cross-links through protein. It can't cross-link DNA to DNA directly, and there are all kind of technical questions associated with this. And I'll also get to this, if I have time, a little bit on. Okay.
So I have a question.
Yeah.
Is any of these interactions like cell type specific?
So these specific interactions are not cell type specific. They're strong, they're ubiquitous, but we found others that are cell type specific.
In the single cell Hi-C?
Sorry?
In the single cell Hi-C?
So no, not in single cell Hi-C. This is just in standard Hi-C.
But I mean, in standard you wouldn't see it, right? Because you would see the average.
You see the average. So this is why if you look, between chromosomes in general, it looks like you saw. It's like a desert. But if you know where to look in the desert, then you can find it.
Which also makes sense because of the volume. So it's the probability that something is on the outside and will interact with the rest of the things that are...
Yeah, but it's a good question regarding function of these things, because if they're low frequency, then what is the function? What can the function be? Could it be functional? Maybe it's a stochastic phenomena that you want just in some of the cells, right? So a very interesting discussion to make about this. But we can talk some more. So there's a very nice example with olfactory receptors that was published by Stavros Lomvadas in Nature. that they found. And there you actually need a stochastic selection of an olfactory receptor for the smell on neurons. But we can talk about it later. Okay, the next thing you see is this strong diagonal, kind of distance-dependent interaction that essentially things which are near each other tend to interact a lot. Things that are far away tend to interact less. And okay, so I'll just say this seems kind of uninteresting. The way that people usually look at it is with these plots. Sometimes it's called a scaling plot or genomic distance. Essentially, you have genomic distance here on the x-axis, and you have contact probability on the y-axis. Okay? So this is like every point here is like the average of a diagonal. You're taking the average of a diagonal. And what you see is that essentially points, loci, which are close to each other in the genome, tend to interact a lot and less so far away. Okay? So you could say, okay, why is this interesting? First of all, this physically, intuitively, it makes sense. So what does it represent? It represents the fact that if you just take 2 points on a polymer, they tend to interact a lot, just by chance. If you take 2 things far away, it's just like they're randomly floating in space. They're not going to meet each other a lot. Okay? So this is the intuition. And I'm not going to go too much into details here, but you can plot these on a log-log plot. So both of these are in log now. When you do that, this is from the first Haifa. People saw that this is kind of almost follows a line where the slope is -1. Okay? So you can describe it this way. The probability of interaction is proportional to the distance between the loci, I and j, to the power of alpha, which is the slope, which is -1. Okay? Now, what does this mean? So for polymer physicists, actually, these are kind of the things that they're interested in. This is the kind of thing that they look and they can tell you things about the nature of the polymer. They can do simulations and try to see if their polymers give this type of slope. And of course, you're representing the whole interaction map with one number, so it's an oversimplification, huge oversimplification, but sometimes you can learn something. So for example, in this case, essentially you would expect a random polymer, you would expect a slope like this of -1.5. In the initial Haifa paper, they found the slope of -1, and this suggested a specific kind of structure called the fractal globule, in which actually the chromosomes are not mixed and you don't have knots, it makes sense when you want to later on separate chromosomes. Okay, maybe. Okay, so the point is that this can still be somewhat informative, not an enhancer promoter and so on, but okay. Another thing to say, you know what, I'm going to maybe skip this. Okay. There's just another example. I'll show you just to say in one sentence. If you follow now, you look at, I'm not going to say much about dynamics, but if you follow through cell cycle, now you synchronize a population of cells, you actually see that in the mitotic chromosome, you lose all these kind of structures which we didn't talk about yet, and you get this kind of smooth thing, and then they're reestablished. Okay, and then you can, look at the slope and analyze it and come up with all kind of what might the structure be. But we're going to skip that. Okay. The next thing I want to talk about is, so we're going smaller and smaller and smaller in scale. Okay. The next thing is genomic compartments. Okay, so what is genomic compartments? It's essentially this kind of checkerboard pattern that we see, kind of distorted checkerboard. You see it's kind of high, low, high, low, high, low, and here, and so on. Okay, so you see all these kind of rectangles and squares. Okay, so this was observed already in the first Haifa experiments. And the interpretation of this was as follows. So the interpretation was that there are two types of chromatin. One is called A, was called A compartment, the other is called B. And A tends to interact with A, B tends to interact with B, and then you will get a pattern that looks like this. Okay? Now if I say two types of chromatin, what do you think about? Active and inactive, right? Euchromatin, heterochromatin, right? So you're right. So it turns out these correlate with active. A is active, so it's easy to remember, and B is inactive chromatin in terms of their correlations. And you can see that they are correlated with all kind of marks, histone marks, right? These two are heterochromatin. You have different types of heterochromatin, right? And you see correlations. You see correlation, for example, the active chromatin is correlated with genes, right? With DNA's accessibility, and so on. Okay, so you have all these different things correlated with the active chromatin. And these things now can change between cell types. And this makes sense, right? Because we know that between cell types, different regions can change their chromatin state, at least regions which are not constitutive. And you can see, for example, let's look, for example, in this map, that you can see this stripe. There's a change in this stripe, okay? And this also reflects a change here in the underlying histones, okay? So there is some correlation between these things, and they're cell type specific. Okay. So what drives these things together? So we said there's active chromatin and inactive chromatin. What causes active to meet active and inactive to meet inactive? Phase separation. Phase separation, good. One is phase separation, okay? So you have pockets of activity in the middle of inside of the nucleus. You have these pockets, and the other is attachment to the nuclear lamina. Okay, so usually you have these attachment to lamins. So you have around here, you have a heterochromatin. You also have next to the nucleolus, by the way. And with liquid-liquid phase separation, you have pockets of activity inside the nucleus. Okay, so this also, if you remember previously, we talked about radial positioning. So there's this general rule that people have that when you're inside the nucleus, a gene, for example, is inside, it's usually active. When it's near the nuclear lamina, it's inactive. Okay. So I want to briefly show you something, again, going to just a tiny bit of research. So, you know, the first law of genomics is that everything is correlated with GC content, right? This is all, you know it, right? It's either correlated or anti-correlated, but this is always the case. So the same here, right? It's correlated with GC content. You have all these different things. So you have A compartment, you know, it's correlated with gene content, accessible chromatin, active chromatin marks, euchromatin, and radial positioning. How do you go beyond this? So we were actually lucky that we had an experiment that failed miserably. That we took a sperm protein. I won't tell you the whole story. If you want to ask me later. We were trying to study sperm chromatin. It's really hard to do. So we said, well, you know what? Let's take a somatic cell and try to make sperm chromatin in it. So we took a sperm chromatin protein and expressed it in this cell. And it didn't work at all. But what it did was something really cool. So let me show you. Okay, so we express this thing called protamine, and what we saw was, this is after 24 hours, we saw these blobs of protamine after 48 hours, and this is after 72 hours. And look what happened to the histones and the DNA So essentially what happened is that this protein kind of filled up all the center and pushed all the genome to the periphery. Okay? So huge change to the genome, right? You take all of the genome, now it's, if you think in terms of, let's say, radial positioning, now everything is close to the nuclear lamina, right? We said, well, you know, that really doesn't look anything like sperm chromatin, but it's a really interesting perturbation, right? Because now, let's look what happens to the high C. So what do you think happened to the high C? Nothing happened to the high C. Well, almost nothing. Okay, so actually you do see small changes. I won't have time to talk about it. But if you look just by eye, it looks really, really similar. So the up, this is the kind of the wild type, or where you don't have the protamine expressed, this is the bottom. So you have this huge change, but it's not reflected in the interactions. It's not because we don't think it's a problem with the experiment. We think that it's because you're measuring really different things, okay? One is pairwise interactions, right? That maybe they can be held together even if you move things along, right? And you know, nuclei in physiological stances, sometimes they need to, you know, cells need to go through all kind of thin places in the tissue and so on, so they need to maintain stuff, right? But to us, this is really interesting, and we're still trying to figure out what it means, but it's really important for you to remember that really microscopy and high C measure really different things, and they're complementary. So just by looking at the high C, you might be missing a really, really big change. Okay? Good. This is on bioRxiv. If you want, you can also look at it. Though we're making many interesting experiments for the revision, so maybe wait for that. Okay, how are we on time? 3:30. I think we're going to keep going and finish the patterns. It's not that long, so stay strong. Everyone still, do you want a break or get up, shout something? Okay. The next pattern is something called topologically associating domains. Okay, so now we're going to look at the matrix a little different. You probably saw this. You can, because the matrix is symmetric, we're going to look at just half of it. You're going to rotate it 45 degrees. We're going to look at it this way. Okay, so now we're looking at the interaction data this way. If we look at two loci, we can look at their intersection, and that's how frequently they interact. So back again, this is more than a decade ago, people saw these kind of squares and smaller squares along the diagonal, and these were called TADs. Okay, so what does it mean? It means that essentially if you have two loci which are in the same triangle, within the same TAD, they might interact more frequently than these two loci, which are in different TADs, just because they're in this same area. This is why the kind of the name domain makes sense, okay? Because things inside this thing are going to interact frequently, and things in different domains are not going to interact frequently, okay? So these were found to be largely conserved both between cell types and even in synthetic regions between species. And you even find similar types of structures in a yeast and even in bacteria. Okay? I'm going to very briefly I'm not going to show, but really, if you want to read a really, really cool paper, I really recommend this one. By Lupianez in Cell, what they found, I'll tell you in a few words, is they looked at all kind of rearrangements in the developmental locus. Okay, this is in humans. In patients, they found that you have all these kind of phenotypes associated with the fingers, developmental phenotypes like polydactyly. what was interesting is that the rearrangements that they found, they weren't deleting, well, they were sometimes deleting this gene FF4, but it's not really important. It doesn't do anything. But you can get all kind of inversions, deletions of stuff kind of in between genes. It doesn't seem like it's deleting genes, and it doesn't seem like it's deleting regulatory regions. But still, it has a huge outcome. It turned out that what's happening, and this was just a bit after the discovery of TADs, that what is changing here is the TAD structure. Okay? So the TADs, according to this view, today it's a little more complicated, but the original view is that this TAD is like a domain in which you have interactions, things can meet each other. It's like a regulatory microenvironment. Okay? So if you put a gene there, it's going to sense the enhancers within the domain, but not in another domain. But if you delete the boundary of this domain, then you're going to have effects by enhancers in the other domain. Okay? So what happened was in that in each of these cases, you had suddenly an interaction of the gene with this domain. It acquired this expression pattern in development, and you got this phenotype. Okay. So now, I'm not saying anything about what the mechanism is. For this, I'm going to go to the last pattern. Okay, so we're almost done. Hold in there. And I'm going to connect it to the last pattern because all of this stuff seems, okay, well, it's correlated with, chromatin, whatever. You're saying, but I'm interested in kind of enhancer promoter, gene regulation, and so on. Okay, so where is all the enhancer promoter stuff? Okay. So as the Hi-C maps got better, we got better resolution. You could sequence more because of developments in next generation sequencing. Then people started to see these dots. Okay, so they're often called loops. I don't really like this term. So why is it called a loop? Well, if you think about it, you say, it's an interaction between this and this. Right, that's consistent, right? Let me kind of grab a cable here without ruining anything. Never mind. Okay, you can imagine. Here, good. Okay, so you have these two, right, in a loop. These two are touching each other. The rest, not so much. This is not touching so much. So you just have the base essentially held together, right? So you say, okay. These dots, they're probably, this is enhancer promoter interactions, right? So you have kind of a enhancer, promoter, good. It turns out that they're not actually. So they're actually often at the tips of these TADs, and you have a binding of this protein called CTCF at the boundaries, okay? And you actually, if you look in Hi-C maps, you are going to miss many, many enhancer promoter interactions, even though people know and they're being functionally validated and so on. Okay? So it turns out, and okay. I'll tell you, this is, again, a very, very interesting story that we don't have that much time to talk about. But, so it turns out, there's Dimitri here, right? That he can probably tell you more about this. I think this is exactly what he works on. So you have these complexes. I'll just show you the movie. Maybe it's the best. So look, you can see one of these complexes in action. Whoop. These are SMC complexes that they land. I'll show you the bottom one, maybe this one. It's easier to see. They land on the DNA, and they start to do this thing. Okay? So they start to extrude the loop. It's called loop extrusion. Okay? They do this until either they fall off, or they bump into a boundary, which is this CTCF, or they bump into another machine. Okay? And through a very, very nice set of papers, people have shown, and you saw it here also, these people have actually been able to film this. This is on a slide. This is not on a cell. You have the DNA attached here. There's a flow over here, and you can see the loop extruder fall here and start. You will see the loop start to extend as this happens. Okay. This is a real cool story, and it's probably what causes these structures. Okay, so that's the extruded loop. Okay. Okay, so actually what's happening here is that you have extruders falling here. They're starting to move until they either fall off or get stuck. And because of that, you don't have interactions over the boundary because the loop extruders can't pass. Okay, so this is the explanation of TADS. So the last thing is now, where are all the enhancer-promoter interactions? Okay, so it's really disappointing, right? So people sequenced more and more in Hi-C, but they didn't see enhancer-promoter interactions. And I have to say that I by myself was kind of skeptical whether enhancer-promoter interactions as they appear in the textbook actually exist. Okay? How do we know that they exist? Did you ask yourself, right? Everyone knows this cartoon, right, where they touch each other. How do you show that? I mean, you can show maybe one, right, with microscopy. It's also actually quite advanced. How do you test that? Okay? So when I saw this map, I was actually quite skeptical. And it turns out it's not a sequencing depth problem. So remember what we said was one way to improve your sequencing depth? That was looking just at a subset of interactions, of loci. So what people have done, for example, is to select a subset of loci in the following way. So we have this standard experiment, but now we take our pool of interactions and we use an antibody for a specific histone mark that marks active promoters, and we pull down only interactions of active promoters. Okay? And then we get a very, very high resolution map of only active promoter interactions. Okay? So people started doing this in experiments like PlaqueSeq and HiChip and so on. And this is what it looks like. So we're looking at the oncogene MYC, which you probably know, and see its interactions of this gene with the rest of the region. Okay? And if you look at the known enhancers, these are all kind of validated enhancers. You see kind of not what you imagine, right? You would imagine to see peaks there, right? If there's a loop, you would expect to see a very stable interaction in each of these positions, and you actually don't see that. So even if you kind of sequence very deeply, here you barely see anything, here you see something but kind of very shallow. So where are the enhancer-promoter interactions? Right? Maybe you can think of, and this has been discussed, there are other models, right, of enhancer-promoter action during transcription. This is the simplest one. There are others where you have a stable loop before transcription, okay? Maybe the loop is held before. And there are other ones like touch and go, that you just touch once, you don't maybe need a stable interaction, and then the gene fires and so on. And there are also some microscopy evidence showing, for example, this is a kind of extreme case, where you actually have an enhancer promoter that when they are activated, the gene is activated, they actually move farther apart from each other. Okay, so it's unclear what really is happening. Whether this is biological or technical, and I'm almost done, so don't worry. And then, so Arkady joined my lab from Sergei Razin and Alexei Gavrilov's lab, and he developed this new version of a C experiment where he took ideas from different experiments and put them together. And it turned out that the little details, in the end it was things like the detergent you use and so on, really changed the picture. Okay, so what he did was as follows. So first of all, he used this antibody selection, so you measure only interactions of active promoters. And the second thing he did was, maybe the main thing, was using, for the restriction enzyme, micrococcal nuclease. This was not our invention, but this is used in an improved version of Hi-C called micro-C, which you will also see in different places. Actually, now in the data we're going to use, we're going to look at micro-C data. Essentially, it has some differences, but it just gives you higher resolution. Okay? Long story short, this is how the data looks with Plaxic, and this is how it looks with Arkady's experiment, which is called mChIP-C. And this is maybe how I imagined enhancer-promoter interaction should look like. Okay, so it turned out that actually enhancer-promoter interactions, and you can read the paper, but you can see then validate this on a large scale, and you see that actually most validated enhancer-promoter pairs, if you use this experiment, in 60 to 70%, you do see a strong interaction between them. Okay, so the point here is that even with these experiments, you have to be careful not to treat them as the truth. Okay, and so as was mentioned here, in terms of interchromosomal interactions, there might be things that the experiment doesn't measure correctly. So the same goes for enhancer-promoter interactions. Remember this, if you're looking at Hi-C, it's going to be very, very difficult to look at enhancer-promoter interactions, and even in micro-C. Okay, so yes, so this is the end of the story, and okay. So I think, let's see, how much time? I think it's a good time for a break now.